Dominance in genetics is a relationship between alleles of one gene, in which one allele is expressed over a second allele at the same locus. The first allele is dominant and the second allele is recessive. For genes on an autosome, the alleles and their associated traits are autosomal dominant or autosomal recessive. Dominance is a key concept in Mendelian inheritance and classical genetics. Often the dominant allele codes for a functional protein whereas the recessive allele does not. A classic example of dominance is the inheritance of seed shape in peas. Peas may be round or wrinkled. In this case, three combinations of alleles are possible, RR, RR, and RR. The RR individuals have round peas and the RR individuals have wrinkled peas. In RR individuals the R allele masks the presence of the R allele, so these individuals also have round peas. Thus, allele R is dominant to allele R, and allele R is recessive to allele R. More generally, where a gene exists in two allelic versions, three combinations of alleles are possible, AA, OR, and OR. If AA and OR individuals show different forms of some trait, and OR individuals show the same phenotype as AA individuals, then allele A is said to dominate or be dominant to or show dominance to allele, and A is said to be recessive to A. Dominance is not inherent to an allele. It is a relationship between alleles. One allele can be dominant over a second allele, recessive to a third allele, and codominant to a fourth. Dominance differs from epistasis, a relationship in which an allele of one gene affects the expression of another allele at a different gene. Background. Diploid/haploid, most familiar animals and some plants have paired chromosomes, and are described as diploid. They have two versions of each chromosome, one contributed by the female parent in her ovum, and one by the male parent in his sperm, which are joined at fertilization. The ovum and sperm cells have only one copy of each chromosome and are described as haploid. Production of haploid gametes occurs through a process called meiosis. Chromosomes, genes, and alleles. Each chromosome of a matching pair is structurally similar to the other, and each member of a homologous pair has a very similar DNA sequence. The DNA in each chromosome functions as a series of discrete genes that influence various traits. Thus, each gene also has a corresponding homologue, which may exist in different versions called alleles. The alleles at the same locus on the two homologous chromosomes may be identical or different. In popular use, gene and allele are often used interchangeably. This produces misunderstandings. Properly, gene refers to a hereditary unit, ordinarily at a fixed position on a chromosome, that influences a particular trait. Genes are sequences of DNA. Allele refers to any of the many particular versions of a gene that may be present in a population of individuals from a particular species, at a particular locus. For example, it is inaccurate to say this pea plant has a pair of wrinkled genes, and it is more accurate to say, this plant has two W alleles for the gene that influences seed shape, and will therefore produce wrinkled peas. Consider also the example of blood type in humans. Near the long arm of chromosome 9 appears a gene that determines whether an individual will be blood type A, B, or O. There are three different alleles that could be present at this locus, but only two can be present in any individual, one inherited from their mother and one from their father. Homozygous, heterozygous, if two alleles of a given gene are identical, the organism is called a homozygote and is said to be homozygous with respect to that gene. If instead the two alleles are different, the organism is a heterozygote and is heterozygous. The genetic makeup of an organism either at a single locus or over all its genes collectively, is called its genotype. The genotype of an organism directly and indirectly affects its molecular, physical, and other traits, which individually or collectively are called its phenotype. At heterozygous gene loci, the two alleles interact to produce the phenotype. One important kind of allele interaction is that described by Mendel, now called Mendelian, in which the heterozygous individual has the appearance phenotype caused by one allele when homozygous, called dominant, and not the appearance phenotype caused by the other allele. The alleles in this case are said to be dominant and recessive to each other, respectively. Dominance, complete dominance, incomplete dominance, The effect of one allele in a heterozygous genotype completely masks the effect of the other. The allele that masks the other is said to be dominant to the latter, and the allele that is masked is said to be recessive to the former. Complete dominance therefore means that the phenotype of the heterozygote is indistinguishable from that of the dominant homozygote. A classic example of dominance is the inheritance of seed shape in peas. Peas may be round or wrinkled. In this case, three combinations of alleles are possible: RR and RR homozygous and RR is heterozygous. The RR individuals have round peas and the RR individuals have wrinkled peas. In RR individuals the R allele masks the presence of the R allele, so these individuals also have round peas. Thus, allele is dominant to allele R, and allele is recessive to allele R, incomplete and semi-dominance. Incomplete dominance occurs when the phenotype of the heterozygous genotype is distinct from and often intermediate to the phenotypes of the homozygous genotypes. For example, the snapdragon flower color is either homozygous for red or white. When the red homozygous flower is paired with a white homozygous flower, the result yields a pink snapdragon flower. The pink snapdragon is the result of incomplete dominance. A similar type of incomplete dominance is found in the 4 o'clock plant wherein pink color is produced when true-bred parents of white and red flowers are crossed. In quantitative genetics, where phenotypes are measured and treated numerically, if a heterozygote's phenotype is exactly between that of the two homozygotes, the phenotype is said to exhibit no dominance at all, that is, dominance exists only when the heterozygote's phenotype measure lies closer to one homozygote than the other. When plants of the F1 generation are self-pollinated, the phenotypic and genotypic ratio of the F2 generation will be 1:1 for both generations. Codominance. Codominance occurs when the contributions of both alleles are visible in the phenotype. For example, in the ABO blood group system, chemical modifications to a glycoprotein on the surfaces of blood cells are controlled by three alleles, two of which are codominant to each other and dominant over the recessive I at the ABO locus. The IA and IB alleles produce different modifications. The enzyme coded for by IA adds an N-acetylgalactose to the membrane-bound H antigen. The IB enzyme adds a galactose. The I allele produces no modification. Thus IA and IB alleles are each dominant to I. Another example occurs at the locus for the beta-globin component of hemoglobin, where the three molecular phenotypes of HbA-HbA, HbA-HbS, and HbS-HbS are all distinguishable by protein electrophoresis for most gene loci at the molecular level. Both alleles are expressed codominantly, because both are transcribed into RNA. Codominance, where allelic products coexist in the phenotype, is different from incomplete or semi-dominance, where the quantitative interaction of allele products produces an intermediate phenotype. For example, in codominance, a red homozygous flower and a white homozygous flower will produce offspring that have red and white spots. When plants of the F1 generation are self-pollinated, the phenotypic and genotypic ratio of the F2 generation will be 1:1. These ratios are the same as those for incomplete dominance. Again, note that this classical terminology is inappropriate. In reality such cases should not be said to exhibit dominance at all. Which trait is dominant? By definition, the terms dominant and recessive refer to the genotypic interaction of alleles in producing the phenotype of the heterozygote. The key concept is genetic, which of the two alleles present in the heterozygote is expressed, such that the organism is phenotypically identical to one of the two homozygotes. It is sometimes convenient to talk about the trait corresponding to the dominant allele as the dominant trait, and the trait corresponding to the hidden allele as the recessive trait. However, this can easily lead to confusion in understanding the concept as phenotypic. For example, to say that green peas dominate yellow peas confuses inherited genotypes and expressed phenotypes, and will subsequently confuse discussion of the molecular basis of the phenotypic difference. Dominance is not inherent. One allele can be dominant to a second allele, recessive to a third allele, and codominant to a fourth. Dominance is unrelated to the nature of the phenotype itself, that is, whether it is regarded as normal, or abnormal, standard, or non-standard, healthy, or diseased, stronger, or weaker, or more or less extreme. A dominant allele may account for any of these trait types. Other distinctions are made between the gene locus, the alleles at that locus, and the phenotype the alleles produce. Nomenclature: in genetics, symbols began as algebraic placeholders. When one allele is dominant to another, the oldest convention is to symbolize the dominant allele with a capital letter. The recessive allele is assigned the same letter in lowercase. In the P example, once the dominance relationship between the two alleles is known, it is possible to designate the dominant allele that produces a round shape by a capital letter symbol R, and the recessive allele that produces a wrinkled shape by a lowercase symbol r. The homozygous dominant, heterozygous, and homozygous recessive genotypes are then written AR, AR, and AR, respectively. It would also be possible to designate the two alleles as W and W, and the three genotypes WW, WW, and WW, the first two of which produced round Ps and the third wrinkled Ps. Note that the choice of R, or W as the symbol for the dominant allele does not prejudge whether the allele causing the round, or wrinkled phenotype when homozygous is the dominant one. In most cases, genes have several alleles. If multiple dominant alleles exist, they may be designated with different superscripts over the uppercase letter, for example X. If multiple recessive alleles exist, they are designated with different superscripts on the lowercase a, for example X. To indicate the two alleles are codominant, they are both written in uppercase, with a superscript to indicate the different alleles. Rules of genetic nomenclature have evolved as genetics has become more complex. Committees have standardized the rules for some species, but not for all. Rules for one species may differ somewhat from the rules for a different species. Relationship to other genetic concepts, multiple alleles. Although any individual of a diploid organism has at most two different alleles at any one locus, most genes exist in a large number of allelic versions in the population as a whole. If the alleles have different effects on the phenotype, sometimes their dominance interactions with each other can be described as a series. For example, coat color in domestic cats is affected by a series of alleles of the TYR gene. The alleles C, CB, CS, and CAR produce different levels of pigment and hence different levels of color dilution. The C allele is completely dominant over the last three and the CAR allele is completely recessive to the first three. Autosomal versus sex-linked dominance. In humans and other mammal species, sex is determined by two sex chromosomes called the X chromosome and the Y chromosome. Human females are typically 20. Males are typically XY. The remaining pairs of chromosome are found in both sexes and are called autosomes. Genetic traits due to loci on these chromosomes are described as autosomal, and may be dominant or recessive. Genetic traits on the X and Y chromosomes are called sex-linked, because they are linked to sex chromosomes, not because they are characteristic of one sex or the other. In practice, the term almost always refers to X-linked traits and a great many such traits are not affected by sex. Females have two copies of every gene locus found on the X chromosome, just as for the autosomes, and the same dominance relationships apply. Males however have only one copy of each X chromosome gene locus, and are described as homozygous for these genes. The Y chromosome is much smaller than the X, and contains a much smaller set of genes, including, but not limited to, those that influence maleness, such as the SRY gene for testis determining factor. Dominance rules for sex-linked gene loci are determined by their behavior in the female. Because the male has only one allele, that allele is always expressed regardless of whether it is dominant or recessive. Epistasis. Epistasis, epiplastasis equals to sit on top is an interaction between alleles at two different gene loci that affect a single trait, which may sometimes resemble a dominance interaction between two different alleles at the same locus. Epistasis modifies the characteristic 9:3:1 ratio expected for two non-epistatic genes. For two loci, 14 classes of epistatic interactions are recognized. As an example of recessive epistasis, one gene locus may determine whether a flower pigment is yellow or green, while another locus determines whether the pigment is produced or not. In a BB plant, the flowers will be white, irrespective of the genotype of the other locus as AA, or. The BB combination is not dominant to the A allele, rather, the B gene shows recessive epistasis to the A gene, because the B locus when homozygous for the recessive allele suppresses phenotypic expression of the A locus. In a cross between two all plants, this produces a characteristic 9:3:4 ratio, in this case of yellow green or white flowers. In dominant epistasis, one gene locus may determine yellow or green pigment as in the previous example, AA and all were yellow, and all were green. A second locus determines whether a pigment precursor is produced or not. Here, in a DD or DD plant, the flowers will be colorless irrespective of the genotype at the A locus, because of the epistatic effect of the dominant D allele. Thus, in a cross between two odd plants, 3/4 of the plants will be colorless, and the yellow and green phenotypes are expressed only in DD plants. This produces a characteristic 12:3:1 ratio of Whittier yellow-green plants. Supplementary epistasis occurs when two loci affect the same phenotype. For example, if pigment color is produced by CC or CC but not CC, and by DD or DD but not DD, then pigment is not produced in any genotypic combination with either CC or DD. That is, both loci must have at least one dominant allele to produce the phenotype. This produces a characteristic 9:7 ratio of pigmented to unpigmented plants. Complementary epistasis in contrast produces an unpigmented plant if and only if the genotype is CC and DD, and the characteristic ratio is 15:1 between pigmented and unpigmented plants. Classical genetics considered epistatic interactions between two genes at a time. It is now evident from molecular genetics that all gene loci are involved in complex interactions with many other genes, and that this creates epistatic interactions that are much more complex than the classic two-locus models. Hardy-Weinberg principle. The frequency of the homozygous state can be estimated using the Hardy-Weinberg formula. This formula applies to a gene with exactly two alleles and relates the frequencies of those alleles in a large population to the frequencies of their three genotypes in that population. For example, if P is the frequency of allele A, and Q is the frequency of allele, then the terms P2, 2PQ, and Q2 are the frequencies of the genotypes AA, or respectively. Since the gene has only two alleles, all alleles must be either A or A and P plus Q equals 1. Now, if A is completely dominant to it, then the frequency of the carrier genotype or cannot be directly observed, however it can be estimated from the frequency of the recessive trait in the population, since this is the same as that of the homozygous genotype, or that is the individual allele frequencies can be estimated, Q equals a SHF, or P equals 1 a euro Q. And from those the frequency of the carrier genotype can be derived, F, or, equals 2PQ. This formula relies on a number of assumptions and an accurate estimate of the frequency of the recessive trait. In general, any real-world situation will deviate from these assumptions to some degree, introducing corresponding inaccuracies into the estimate. If the recessive trait is rare, then it will be hard to estimate its frequency accurately, as a very large sample size will be needed. Molecular mechanisms. The molecular basis of dominance was unknown to Mendel. It is now understood that a gene locus includes a long series of bases or nucleotides of deoxyribonucleic acid at a particular point on a chromosome. The central dogma of molecular biology states that DNA makes RNA makes protein, that is, that DNA is transcribed to make an RNA copy, and RNA is translated to make a protein. In this process, different alleles at a locus may or may not be transcribed, and if transcribed may be translated to slightly different versions of the same protein. Proteins often function as enzymes that catalyze chemical reactions in the cell, which directly or indirectly produce phenotypes. In any diploid organism, the DNA sequences of the two alleles present at any gene locus may be identical or different. Even if the gene locus is heterozygous at the level of the DNA sequence, the proteins made by each allele may be identical. In the absence of any difference between the protein products, neither allele can be said to be dominant. Even if the two protein products are slightly different, it is likely that they produce the same phenotype with respect to enzyme action, and again neither allele can be said to be dominant. Loss of function and haploinsufficiency. Dominance typically occurs when one of the two alleles is non-functional at the molecular level, that is, it is not transcribed or else does not produce a functional protein product. This can be the result of a mutation that alters the DNA sequence of the allele. An organism homozygous for the non-functional allele will generally show a distinctive phenotype, due to the absence of the protein product. For example, in humans and other organisms, the unpigmented skin of the albino phenotype results when an individual is homozygous for an allele that prevents synthesis of the skin pigment protein melanin. It is important to understand that it is not the lack of function that allows the allele to be described as recessive, this is the interaction with the alternative allele in the heterozygote. Three general types of interaction are possible. In the typical case, the single functional allele makes sufficient protein to produce a phenotype identical to that of the homozygote. This is called haploinsufficiency. For example, suppose the standard amount of enzyme produced in the functional homozygote is 100%, with the two functional alleles contributing 50% each. The single functional allele in the heterozygote produces 50% of the standard amount of enzyme, which is sufficient to produce the standard phenotype. If the heterozygote and the functional allele homozygote have identical phenotypes, the functional allele is dominant to the non-functional allele. This occurs at the albino gene locus. The heterozygote produces sufficient enzyme to convert the pigment precursor to melanin, and the individual has standard pigmentation. Less commonly, the presence of a single functional allele gives a phenotype that is not normal but less severe than that of the non-functional homozygote. This occurs when the functional allele is not haplosufficient. The terms haploinsufficiency and incomplete dominance are typically applied to these cases. The intermediate interaction occurs where the heterozygous genotype produces a phenotype intermediate between the two homozygotes. Depending on which of the two homozygotes the heterozygote most resembles, one allele is said to show incomplete dominance over the other. For example, in humans the HB gene locus is responsible for the beta chain protein that is one of the two globin proteins that make up the blood pigment hemoglobin. Many people are homozygous for an allele called HbA. Some persons carry an alternative allele called HbS either as homozygotes or heterozygotes. The hemoglobin molecules of HbS homozygotes undergo a change in shape that distorts the morphology of the red blood cells, and causes a severe, life-threatening form of anemia called sickle cell anemia. Persons heterozygous HbA-HbS for this allele have a much less severe form of anemia called sickle cell trait. Because the disease phenotype of HbA-HbS heterozygotes is more similar to but not identical to the HbA-HbA homozygote, the HbA allele is said to be incompletely dominant to the HbS allele. Rarely, a single functional allele in the heterozygote may produce insufficient gene product for any function of the gene, and the phenotype resembles that of the homozygote for the non-functional allele. This complete haploinsufficiency is very unusual. In these cases the non-functional allele would be said to be dominant to the functional allele. This situation may occur when the non-functional allele produces a defective protein that interferes with the proper function of the protein produced by the standard allele. The presence of the defective protein dominates the standard protein, and the disease phenotype of the heterozygote more closely resembles that of the homozygote for two defective alleles. Note that the term dominant is often incorrectly applied to defective alleles whose homozygous phenotype has not been examined, but which cause a distinct phenotype when heterozygous with the normal allele. This phenomenon occurs in a number of trinucleotide repeat diseases. For an example and more details see Huntington disease. Dominant negative mutations. Many proteins are normally active in the form of a multimer, an aggregate of multiple copies of the same protein, otherwise known as a homomultimeric protein or homoligomeric protein. In fact, a majority of the 83,000 different enzymes from 9,800 different organisms in the Brenda enzyme database represent homoligomers. When the wild-type version of the protein is present along with a mutant version, a mixed multimer can be formed. a mutation that leads to a mutant protein that disrupts the activity of the wild-type protein in the multimer is a dominant negative mutation. A dominant negative mutation may arise in a human somatic cell and provide a proliferative advantage to the mutant cell, leading to its clonal expansion. For instance, a dominant negative mutation in a gene necessary for the normal process of programmed cell death in response to DNA damage can make the cell resistant to apoptosis. This will allow proliferation of the clone even when excessive DNA damage is present. Such dominant negative mutations occur in the tumor suppressor gene p53. The p53 wild-type protein is normally present as a 4-protein multimer. Dominant negative p53 mutations occur in a number of different types of cancer and precancerous lesions. Dominant negative mutations also occur in other tumor suppressor genes. For instance, 2 dominant negative germline mutations were identified in the ataxia-telangiectasia mutated gene, which increases susceptibility to breast cancer. Dominant negative mutations of the transcription factor CP+or- can cause acute myeloid leukemia. Inherited dominant negative mutations can also increase the risk of diseases other than cancer. Dominant negative mutations in pyroxene proliferator activated receptor gamma are associated with severe insulin resistance, diabetes mellitus and hypertension. Dominant negative mutations have also been described in organisms other than humans. In fact, the first study reporting a mutant protein inhibiting the normal function of a wild-type protein in a mixed multimer was with the bacteriophage T4 tail fiber protein GP37. Mutations that produce a truncated protein rather than a full-length mutant protein seem to have the strongest dominant negative effect in the studies of p53, ATM, CP+or-, and bacteriophage T4 GP37. Dominant and recessive genetic diseases in humans. In humans, many genetic traits or diseases are classified simply as dominant, or recessive. Especially with respect to so-called recessive diseases, this can oversimplify the underlying molecular basis and lead to misunderstanding of the nature of dominance. For example, the genetic disease phenylketonuria results from any of a large number of alleles at the gene locus for the enzyme phenylalanine hydroxylase. Many of these alleles produce little or no PAH, as a result of which the substrate phenylalanine and its metabolic byproducts accumulate in the central nervous system and can cause severe intellectual disability if untreated. The genotypes and phenotypic consequences of interactions among 3 alleles are shown in the following table. In unaffected persons homozygous for a standard functional allele, PAH activity is standard, and the concentration of phenylalanine in the blood, Phe, is about 60 In untreated persons homozygous for one of the PKU alleles, PAH activity is close to 0, Phe 10 to 40 times standard, and the individual manifests PKU. In the AB heterozygote, PAH activity is only 30% of standard, blood, Phe is elevated 2-fold, and the person does not manifest PKU. Thus, the A allele is dominant to the B allele with respect to PKU, but the B allele is incompletely dominant to the A allele with respect to its molecular effect, determination of PAH activity level. Finally, the A allele is an incomplete dominant to B with respect to Phe, as 60 less than 120 600 Note once more that it is irrelevant to the question of dominance that the recessive allele produces a more extreme, Phe phenotype. For a third allele C, a CC homozygote produces a very small amount of PAH enzyme, which results in a somewhat elevated level of Phe in the blood, a condition called hyperphenylalaninemia, which does not result in intellectual disability. That is, the dominance relationships of any 2 alleles may vary according to which aspect of the phenotype is under consideration. It is typically more useful to talk about the phenotypic consequences of the allelic interactions involved in any genotype, rather than to try to force them into dominant and recessive categories. History. The concept of dominance was introduced by Gregor Mendel. Though Mendel, the father of genetics, first used the term in the 1860s, it was not widely known until the early 20th century. Mendel observed that, for a variety of traits of garden peas having to do with the appearance of seeds, seed pods, and plants, there were two discrete phenotypes, such as round versus wrinkled, yellow versus green seeds, red versus white flowers or tall versus short plants. When bred separately, the plants always produced the same phenotypes, generation after generation. However, when lines with different phenotypes were crossed, one and only one of the parental phenotypes showed up in the offspring. However, when these hybrid plants were crossed, the offspring plants showed the two original phenotypes, in a characteristic 3:1 ratio, the more common phenotype being that of the parental hybrid plants. Mendel reasoned that each parent in the first cross was a homozygote for different alleles, that each contributed one allele to the offspring, with the result that all of these hybrids were heterozygotes, and that one of the two alleles in the hybrid cross dominated expression of the other, A masked A. The final cross between two heterozygotes would produce AA, or offspring in a 1:1 genotype ratio, with the first two classes showing the A phenotype, and the last showing the A phenotype, thereby producing the 3:1 phenotype ratio. Mendel did not use the terms gene, allele, phenotype, genotype, homozygote, and heterozygote, all of which were introduced later. He did introduce the notation of capital and lowercase letters for dominant and recessive alleles, respectively, still in use today. See also, Evolution of Dominance, List of Mendelian traits in humans, Mitochondrial DNA, Punnett Square, References. Online notes for Biology 2250 Euro Principles of Genetics. Memorial University of Newfoundland. Online Mendelian Inheritance in Man Hemoglobin Euro Beta Locus. HBB-141,900 Euro Sickle Cell Anemia, Online Mendelian Inheritance in Man Apoglycosyl Transferase -110,300 Euro Apo Blood Groups, External links, Online Mendelian Inheritance in Man, Autosomal Dominance of Huntington's Disease. Huntington's Disease Outreach Project for Education at Stanford, Examples of Incomplete Dominance.